Welcome back to the Deep Dive. We're here again to sift through complex info and, well, make it stick.
That's the goal, giving you the core insights fast.
And today, we're really getting into the powerhouse of the cell, deep into it.
Absolutely. We're looking at how our bodies take carbohydrates and really squeeze out the energy, the final stages,
Yeah.
the TCA cycle, and oxidative phosphorylation.
Right, these sound complex, maybe a bit intimidating.
They can seem that way, yeah.
Yeah.
But honestly, they're the absolute bedrock of, well, life itself.
So our mission today,
To break them down,
Yeah.
especially for, those of you starting your medical journey, give you a clear map, some useful anchors, metaphors maybe, to help you understand, remember, and recall these pathways.
like a guide to the cell's power grid. Love it.
Exactly.
Okay, let's get started. Most of you listening have probably covered glycolysis, right? Breaking glucose down to pyruvate.
Yeah, the starting point. But
Yeah.
that pyruvate, it's not quite ready for prime time. It needs a pass, like a special ticket to get into the mitochondrial matrix.
The matrix, right, where the main action happens.
Precisely. Think of pyruvate as, raw material arriving at the factory gate, the mitochondrial gate.
Needs processing first.
Needs processing, yeah, into a usable form, which is acetyl-CoA. This happens through a really critical step. It's an oxidative decarboxylation. Meaning it loses electrons, that's oxidation, and it loses a carbon atom as CO2, that's decarboxylation, and it's irreversible, can't go back.
Okay. And what controls this?
A huge enzyme complex, massive thing, called pyruvate dehydrogenase, PDH for short.
PDH. Got it. An anchor for this.
Let's call PDH the toll booth, or maybe the security checkpoint for pyruvate trying to get into the matrix.
I like toll booth. So what does this toll booth need to function?
Good question. It relies heavily on cofactors, many derived from B vitamins. We're talking B1, thiamine, B2, riboflavin, B3, niacin, B5, pantothenic acid.
So deficiencies in B vitamins.
Can really jam up the toll booth, absolutely. Slows everything down right at the gate.
Makes sense. And like a real toll booth, it probably has busy times and slow times. Regulation.
You bet. It has its gas pedals, activators, and brakes inhibitors.
What hits the gas?
Things that signal we need energy. So pyruvate itself, CoA, NAD plus ADP,
Yeah.
even insulin, interestingly, through dephosphorylation.
And the brakes.
Things that signal we've got enough energy. So the product, acetyl-CoA, ATP itself, NADH, even fatty acids, it's like really elegantly controlled.
Okay, this is where it gets really clinically relevant, especially for our listeners heading into medicine.
Mm-hmm.
Let's pause for a thought here. What do you, listening right now, think happens if this PDH toll booth is fundamentally broken?
Yes.
If pyruvate can't get through and become acetyl-CoA, what's the big immediate danger?
Hold that thought. We'll definitely circle back to it.
Okay, so clinically, what if it is broken?
Right. A PDH deficiency. It's a genetic defect, often X-linked, though rare.
And the consequence?
It's the most common cause, actually, of congenital lactic acidosis.
Lactic acidosis. Why?
Well, if pyruvate can't become acetyl-CoA to enter the aerobic pathway, the main energy route, it gets forced down the anaerobic path.
Which produces lactate.
Exactly. Too much lactate builds up, lactic acidosis, and this causes serious neurological problems, muscle issues. It really underlines how vital this single step is. Management often involves diet changes, B vitamin supplements, trying to support what little function might be there.
Wow.
Oh.
Okay, so assuming our pyruvate does get through the PDH toll booth
Pays the toll, becomes acetyl-CoA.
and enters the mitochondrial matrix, now what?
Now we hit stage 3, the TCA cycle, also called the Krebs cycle or the citric acid cycle. Lots of names.
The metabolic roundabout, you called it.
That's my favorite analogy for it, yeah. The metabolic roundabout.
Paint the picture.
Imagine acetyl-CoA, that's our 2-carbon molecule, C2. It's the car entering a roundabout. It merges with a 4-carbon molecule already circulating, oxaloacetate, C4, makes 6. Citrate, a 6-carbon molecule. That's why it's also called the citric acid cycle. It starts with citrate.
And then the roundabout spins.
It spins. The citrate goes through a series of reactions, chemical transformations. Along the way, it releases energy, kicks off 2 molecules of carbon dioxide, that's the exhaust.
Getting rid of carbons.
Exactly, getting back towards the original 4-carbon molecule. And crucially, it regenerates that oxaloacetate at the end. So the roundabout is ready for the next acetyl-CoA to enter. It keeps spinning.
And what's the energy payoff from one spin? One acetyl-CoA.
Key outputs per spin. You get 2 CO2 molecules, like we said. You get 1 GTP, think of it like an ATP, quick energy cache. And most importantly, for the next stage, you generate the electron delivery trucks.
Ah, the NADH and FADH2.
Yep. 3 NADH molecules and 1 FADH2 molecule per spin. These are loaded with high-energy electrons.
And we need to remember, one glucose gives us 2 pyruvates.
Which means 2 acetyl-CoAs.
Yeah.
So for every single glucose molecule you started with way back in glycolysis.
The TCA cycle spins twice.
Exactly. Double everything, double the CO2, double the GTP, double the NADH, double the FADH2.
Are there key control points on this roundabout? Like traffic lights?
Oh, absolutely. There are irreversible steps, mainly the ones catalyzed by citrate synthase, isocitrate dehydrogenase, and alpha-ketoglutarate dehydrogenase. These act like control gates, making sure traffic flows correctly.
And regulated how? Same idea as PDH, energy level.
Pretty much the same principle, yeah. Like the toll booth, it has its own checks and balances. Low energy signals like ADP tend to speed it up, activate it.
Need more energy, spin faster.
Right. And high energy signals like ATP and NADH tend to slow it down, inhibit it. Why spin if the cell's already loaded with energy? It prevents waste.
Makes sense. Now, you also mentioned this cycle is versatile, not just about energy.
Oh, absolutely. It's way more than just burning fuel. It's like a central metabolic hub, a molecular Swiss Army knife, maybe.
Okay. How so?
It's intermediates, the molecules within the cycle. They can be pulled off, siphoned away for building things, anabolic processes.
Like what?
Like making fatty acids or some amino acids, even the heme that's in your hemoglobin. It connects catabolism, breaking down, with anabolism, building up. Really central.
Incredible. Okay, before we move to the final stage, let's revisit that first question we posed.
Ah, yes, the broken PDH toll booth.
If that toll booth is shut down, pyruvate can't become acetyl-CoA. What's the consequence, specifically relating to the TCA cycle and energy?
Well, simply put, if acetyl-CoA can't be made efficiently from pyruvate, the main fuel source for the TCA cycle is cut off.
The roundabout stops spinning, basically.
Or slows dramatically, yeah. So the cell loses its primary, most efficient way of generating those energy carriers, NADH and FADH2, aerobically. And like we said, that pyruvate backs up and gets converted to lactate instead.
Leading to lactic acidosis. A clear sign the main power grid is failing.
Exactly, a dangerous situation.
Okay, that clarifies the link perfectly. So now we have all these electron delivery trucks, and ADH and FADH2 generated from glycolysis, from the PDH reaction, and tons from the TCA cycle.
Yep, loaded up with high-energy electrons.
Where do they go? What's their final drop-off point? And how do they actually generate the huge amounts of ATP we keep talking about? This is the big payoff, right?
This is the big one, stage 4. Oxidative phosphorylation. This is where the vast, vast majority of ATP is made, the real cellular power plant.
And where does this happen? Still in the mitochondria?
Still in the mitochondria, yes, but specifically in the inner mitochondrial membrane. You know how mitochondria diagrams always show those folds inside?
Cristae.
Exactly, the cristae. Those folds massively increase the surface area of that inner membrane because that's where all the machinery for oxidative phosphorylation is embedded.
Okay, and you said it has 2 acts.
2 tightly linked acts, electron transport and ATP synthesis. We need a good metaphor here. Let's go with a hydroelectric power plant.
Okay, I'm listening.
And a power plant.
Act 1, the electron transport chain, the ETC. This is our electron waterfall.
Electron waterfall. So our NADH and FADH2 delivery trucks arrive at the inner membrane. They unload their high-energy electrons onto the first complex in a series of protein complexes embedded in the membrane complexes I through V.
And the electrons flow.
They flow down like water cascading down steps in a waterfall, from one complex to the next, moving to lower and lower energy levels.
And as they fall, energy is released.
Precisely. Energy is released at specific steps, and crucially, what's at the very bottom of the waterfall, the final acceptor for these electrons? Molecular oxygen. O2.
That's why we need to breathe oxygen.
That is exactly why we need oxygen. It's the final electron acceptor. It takes the electrons, combines with some protons, and forms water, H2O. Without oxygen, the whole waterfall backs up. Electrons stop flowing.
Okay, waterfall makes sense. What about the energy released as electrons fall?
That energy is harnessed. Complexes I3 and AV use the energy released from the falling electrons to actively pump protons, hydrogen ions, H+ from the mitochondrial matrix across the inner membrane into the space between the inner and outer membranes, the intermembrane space.
Pumping protons out of the matrix.
Oh, okay. Yeah. So you build up a high concentration of protons in that intermembrane space. It's like water building up behind a dam. You create an electrochemical gradient, a difference in charge and concentration. We call this the proton motive force, or PMF. That's the dam in our analogy. Potential energy, store it up.
Right, like stored water pressure. So that's Act 1.
Yeah.
Build the dam using the electron waterfall. What's Act 2?
Act 2. ATP synthesis, using the energy stored in that dam. This is our hydroelectric turbine.
A turbine, ATP synthesis.
Exactly. Those protons that were pumped out into the intermembrane space, they want to flow back into the matrix down their concentration gradient. But they can't just leak through the membrane.
They need a specific channel.
They need the specific channel. A remarkable protein complex called ATP synthase. It's also called complex V sometimes. As the protons rush back into the matrix through ATP synthase, it's like water rushing through a turbine.
And the turbine spins.
It literally does. The flow of protons causes part of the ATP synthase molecule to rotate, and this mechanical energy drives the chemical reaction, sticking a phosphate group pi onto ADP to make ATP.
Wow. So the flow of electrons powers proton pumping, which creates a gradient, and the flow of protons back down that gradient powers ATP synthesis.
You've got it. Electron transport drives proton pumping, which creates the PMF, which drives ATP synthesis. Tightly coupled.
And the efficiency, you said huge ATP yield.
It's impressive. For each NADH that drops off electrons, the process generates about 2.5 ATPs. For each FADH2, which enters the waterfall a bit lower down, it's about 1.5 ATPs.
So adding it all up from one glucose, glycolysis, PDH, 2 turns of TCA.
You're looking at a grand total of roughly 30 to 32 ATP molecules per glucose. Compare that to the measly 2 net ATP you get from glycolysis alone, anaerobically.
Huge difference. That's why aerobic respiration is so effective.
Absolutely. It powers complex life. And interestingly, it's not 100% efficient at capturing energy as ATP, only about 33–35%.
Where does the rest of the energy go?
Heat. That electron transport process releases a significant amount of energy as heat. And that's not waste. Not really. It's crucial for maintaining our body temperature, especially for us warm-blooded mammals.
Okay, so let's nail down the answer to that second question then. Final destination for NADH, FADH2, and how they make ATP.
Right. The delivery trucks, NADH and FADH2, bring electrons to the electron transport chain, the electron waterfall. Energy from the waterfall pumps protons out, creating the proton dam, the PMF. Then protons flowing back through the hydroelectric turbine ATP synthase drives the production of the vast majority of our ATP.
Electron flow, proton gradient, ADP synthesis. Elegant.
It really is. But that tight coupling, the link between the electron waterfall and the ATP turbine, isn't always absolute. It can be loosened.
Loosened. Why would the body ever want to do that? Less ATP. It seems counterintuitive, given how vital ATP is.
It does seem odd at first glance, doesn't it? Why deliberately make less ATP and just waste that proton gradient?
Yeah, what's the point of uncoupling this amazing system? Any ideas, listeners? Why generate heat instead of ATP?
Think about survival sometimes.
Okay, so the answer to question 3.
The body does sometimes prioritize heat generation over maximum ATP production, particularly for maintaining core body temperature in the cold.
This is where uncoupling comes in, turning the mitochondria into a heat factory.
Exactly, a heat factory. There are natural ways this happens. Special proteins called uncoupling proteins, UCPs, embedded in that inner mitochondrial membrane.
Like UCP1.
Yes. UCP1, also known as thermogenin, is the famous one. It's abundant in brown adipose tissue B-guaiae, or brown fat.
Brown fat. Isn't that important for babies and hibernating animals?
Classically, yes. But adults retain some too, and it's a hot area of research. So when you're cold, your body releases signals like noradrenaline. This triggers fat breakdown in BT, fueling the ETC, the electron waterfall.
Okay, so electrons flow, protons get pumped.
BDT, instead of all those protons flowing back through ATP synthase, the turbine, UCP1 gets activated. It basically opens up an alternative channel, a proton leak.
So protons bypass the turbine.
They bypass the turbine and just flow back into the matrix. The energy stored in that proton gradient, the PMF, isn't used to make ATP. It's just released directly as heat.
Non-shivering thermogenesis, making heat without shivering.
Precisely. It warms you up. And that's why there's interest in maybe activating brown fat or browning white fat for obesity treatment, trying to get the body to burn more calories as heat.
Fascinating. Are there artificial ways to uncouple?
There are, unfortunately. Synthetic chemical uncouplers exist, like dinitrophenol, DNP, or dinitrocresol, DNC. They essentially make the inner membrane leaky to protons.
So same effect. Electrons flow, oxygen is used, but no ATP.
Exactly. Only heat is produced, lots of it, uncontrollably. That's why these substances are incredibly dangerous, highly toxic, even fatal. They short-circuit the whole system.
Right. So that's uncoupling. What about just blocking the system entirely? Inhibition.
That's the other way things go wrong. Inhibition. If you block the electron transport chain itself, the waterfall stops flowing.
No electron flow, no proton pumping.
No proton gradient, no PMF, and therefore no ATP synthesis via ATP synthase. The whole powerhouse grinds to a halt.
What causes that? Lack of oxygen.
Lack of oxygen anaerobic conditions is the most common physiological cause, but specific poisons can do it too, targeting different complexes in the ETC.
Like cyanide.
Exactly. Cyanide and carbon monoxide, CO, are notorious inhibitors of complex 4, the final step where electrons meet oxygen.
So they block the very end of the waterfall.
They do. Others include rotenone, a pesticide which blocks complex 1, and antimycin, an antibiotic which blocks complex 3. They all effectively stall the entire process.
Leading to rapid cell damage and death because the main energy supply is just cut off.
It's incredibly fast and devastating, yes. Shows how dependent we are on this continuous flow.
And besides poisons, genetic issues can also affect this pathway.
Oh, definitely. The complexity means there are many points of potential failure. Diseases of oxidative phosphorylation are a whole category.
Often related to mitochondrial DNA.
Yes, frequently. Mitochondrial DNA, mtDNA, mutates more readily than our nuclear DNA, and since mitochondria are passed down mostly from the mother, these diseases often show maternal inheritance.
Can you give some clinical examples?
Anchors, sure. LHON, Leber hereditary optic neuropathy, often involves mutations affecting complex I, leading to progressive vision loss.
No way.
Or Leys syndrome, a very severe condition usually presenting in infancy with neurological degeneration, often linked to defects in ATP synthase itself, the turbine. And then there are broader syndromes like Mellis mitochondrial encephalopathy, lactic acidosis, and stroke-like episodes which can result from various mitochondrial defects. These really hit home how critical oxidative phosphorylation is, especially for high-energy tissues like the brain and muscles.
Absolutely critical.
Okay.
One last thing before we wrap. We focus heavily on oxidative phosphorylation.
Mm-hmm.
But you mentioned another way cells make ATP earlier, briefly. Substrate-level phosphorylation.
Ah, yes. Good to clarify the distinction. It's worth comparing them directly.
How should we think about them? Direct versus indirect.
That's a great way to put it. Direct versus indirect energy production.
So oxidative phosphorylation is?
Indirect. The energy from oxidizing NADH and FADH2 is indirectly coupled to ATP synthesis via that intermediary, the proton-motive force.
Yeah.
It needs those membrane complexes, the intermitochondrial membrane, and it absolutely requires oxygen. It's the major ATP producer, huge yield, about 33–35% efficient in ATP capture, plus that useful heat.
And substrate-level phosphorylation.
That's direct. Energy coupling happens right there on the enzyme. A high-energy phosphate group is transferred directly from a substrate molecule to ADP, making ATP.
Like in glycolysis.
Exactly like in glycolysis, and also in one step of the TCA cycle. It uses soluble enzymes floating around in the cytosol or the mitochondrial matrix. Doesn't need a membrane, doesn't directly need oxygen, although the pathways occur alongside aerobic metabolism usually.
And the yield.
Much smaller. It's a minor contributor to the total ATP count. Just a few ATPs here and there, but it's actually more efficient in terms of energy capture per reaction, around 60%, with less heat produced.
So an analogy might be, oxidative phosphorylation is like investing your money long-term in a high-yield savings account.
Yeah, the mitochondrial bank, earning lots of interest, ATP.
Well, substrate level is like finding a $20 bill on the street.
Huh, pretty much. Quick cash in your pocket. Useful, happens fast, but it's not going to make you rich. The cell uses both strategies, quick direct payments, and the massive sustained income from the oxidative phosphorylation power plant.
Fantastic. What a detailed tour of the cellular power grid.
It's intricate, isn't it?
Definitely. From the PDH toll booth, letting pyruvate in, to the acetyl-CoA spinning around the TCA roundabout, generating those electron carriers.
And then those carriers powering the electron waterfall, building the proton dam, and driving the ATP turbine of oxidative phosphorylation.
Seeing how it all fits together, how it's regulated, it's quite something.
And understanding it isn't just about, memorizing steps for an exam. It's really about grasping the fundamentals of how life is powered, why B vitamins matter, why oxygen is essential, why certain poisons are so deadly, why these genetic diseases have the effects they do.
It really connects the biochemistry to the clinic.
Absolutely. It's the foundation.
So as you, our listener, continue studying or applying this knowledge, really think about that delicate balance, the regulation, the coupling, the efficiency versus heat. These aren't just lines on a diagram.
They're the whirring machinery, keeping everything running. A heart beating, a nerve firing. Even just thinking requires this constant energy flow.
So, a final thought to leave you with. Based on everything we've discussed, the reliance on oxygen, the potential for lactate buildup, the vulnerability of high-energy tissues, what might be the very first, maybe subtle clinical sign you'd look for if you suspected a patient's entire cellular power grid was starting to falter? Something to ponder.
Good question. That really makes you integrate the pathways.
Exactly.
No.
That's all for this deep dive. Thanks for joining us.
See you next time.